Thanks for coming, everyone. I'm glad we have a good crowd today. So today is a recap. So there's been so much news going on in synbio, really happening every day, but especially in the last month, particularly because there's a lot of conferences going on. So many of you know that the SEED conference was this last week, as well as the SB7.0 conference was a couple weeks ago. Then there's just the general amount of synbio inundation with news. So you will be extremely informed at the end of this meeting. So first we have a synbio newsreel from Adam, and then we're going to go through some highlights of the synbio 7.0 conference, which Isaac's going to do for us, and then we'll have a recap of SEED. So a number of really cool talks there. Northwestern was well represented there as well. So with that, we'll go ahead and start with Adam's talk. And if you haven't yet, go ahead and pick up some pizza as we go. So we're going to actually live stream this presentation, which is kind of cool. So we're trying that out. People who are not here, I guess mostly if you're here, just raise your hand and ask questions. But people can also actually live stream in questions as well. And so Joe is the monitor for that. And so if someone from the interwebs has a question, then they'll have Joe, they'll conduit it through Joe. So anyway. I was not told this. This is now it's pretty exciting, right? You're on the web. Okay. With that, let's thank Adam.
Good hustle.
You might have to. I think you push buttons.
Yeah. Okay, whatever. Hey guys, those of you that we'll just deal with it however it goes. For those of you that don't know me, my name's Adam. I am no longer a first year, I don't think. The Joanne and Lux Labs, you guys can make that call, and I'll be doing the newsreel for today, which of course has to start out recognizing GeneMods has a podcast, and you should all totally go listen to it because it's awesome. So the first episode, Synbio in Space, which is the title with a lot of A's. It's like a Poly A or something, I don't know. Isaac and Jordan did a great job interviewing Professor Chimeric about some of the work that they're doing in that lab. Kind of this space synbio idea of how can we make recombinant proteins in space and what might be some of the useful ideas from that. And there's also at the end a game of wait, wait, don't tell me, essentially, synbio trivia, which is a lot of fun. I wanted to play this. I don't know if this is going to work, but we'll see if we have any volume. Oh, yeah.
These are the voyages of Micro E. coli. It's a 5-year mission to colonize strange new worlds. We're going to talk about synthetic biology in space today with Dr.
So if that's a good enough hook for you, then go ahead and do that. I think the other highlight is learning that Isaac apparently doesn't know what bay means. So if you want to make fun of him for that too. Okay, so like Weston said, there's been a lot of conferences lately, so I'm not going to go too much into detail of things that happened in SB7 or SEED because people that actually know things are going to do that instead. But a lot of the SB7 talks you can find either summaries or the full videos online. So just a couple quick things. The keynote was given by Randall Kirk, who's the CEO of Antigen, which is a group that's been doing a lot of applications of synbio for some interesting new GMO applications. So he was talking a lot about how they can make the salmon that are 3 times as big as the normal salmon, apples that don't brown over time. It's a really cool talk, and he has some choice language about gene drives and biofuels, which I thought was pretty interesting. There was an entire session at the conference devoted to biodiversity, which I think is an issue that don't necessarily hear talked about that much with synthetic biology, but there are people that are doing things. I think a lot of us know about the work that the Church Lab is doing and trying to bring back woolly mammoths. There's also some interesting ideas, for instance, Ryan Phelan, who's talking about how they can engineer coral to be more resistant to bleaching, prevent environmental loss in that way. And then probably the big news that came out of this, there was a note that was sent out to the listservs that Twist Biosciences is partnering with BioBricks to provide 10,000 genes to the public free of charge. You're going to be able to vote on these somehow online. I'm sure Isaac's going to talk a lot more about this later, so I'll let them have the honors. Some more community news. So Boston Bio published this interesting interview with Julie Legault, who is the founder of Amino Labs. Amino Labs is it's a company that's devoted to spreading synbio as a form of outreach to younger kids to try to get them involved in STEM especially. So this is a way of using microbes to make fragrances, colors. She's actually from the MIT Media Lab. That's where she got her start, and then thought that synthetic biology was a really cool way of sort of bringing this science to younger people. And you can actually buy one of these kits where you can make the different fragrances and colors. Community news on the CRISPR side of things, just to make things even more complicated, China sided with the Doudna side on the patent for CRISPR, just like the EU now. So the patent in the EU goes against what's currently the state in the US. So we have a picture of Doudna smiling, and this is the angriest picture I found of Feng online. It turns out if you Google like Feng Zheng frowning, like you just don't get a lot of great things. Also in Doudna news, she's got a book out. It's called The Cracking Creation, and if you are more perceptive than me, then it will take you less than like an hour to figure out that like the letters A, C, T, and G are the ones that are yellow in that title. It's essentially a memoir of the CRISPR saga. The reviews online have been sort of mostly positive, I would say. I haven't gotten the chance to read it or anything yet, but if you want to hear her words about what role she and others had to play in this discovery, then go ahead and check it out. More on Twist. So Microsoft is spent a bunch more money buying more genes from Twist in sort of their goal to encode all of the world's data into DNA. So this was another $10 million arrangement adding on to all the DNA that they bought last year. And I think Catherine talked about some of the work that had happened a couple of GeneMods ago on that front. IDT is also in the news this week. So they launched this new grant program that's specifically devoted to synbio startup companies. So you can apply with what you think your synbio startup will do, and they will reward you in G-blocks. So I did a shout out here for our good friend James, who's leading the Lux Lab later this week. He's always saying, always be cloning. So first place you get 100,000, second place 50,000, third place you're fired. ABCs, right? Okay. On the other side of things, so Bolt Threads, which is one of the companies that's really been involved in getting the spider silk stuff up and running, the yeast brewed or micro brewed spider silk that can potentially go into clothing. They signed this deal with Patagonia, so they're actually making like winter clothing out of this stuff. I was curious what the state is of this. You can actually buy one of these ties, and it sort of looks like spider silk, I guess, but it could like probably pass, and it's only $300. So, like, that's more than I would spend on a tie, as you can tell on the slot, but like, you guys could do it. On the news, on the research side of things, there's an interesting perspective piece in Nature Biotech, primarily focusing on outreach and introducing younger students in high school to STEM, synthetic biology, so programs like iGEM, and they also talk a lot about the Canadian equivalents of iGEM, which are a thing apparently. They look at a bunch of surveys from the people that go to the iGEM jamborees. They also have this kind of interesting bar graph I found of the participation as it's changed over time. So in the blue there is the college programs doing iGEM, and green is high school, which has come up a lot later. What I thought was interesting actually is how the balance has shifted in recent years toward more Asian programs that are participating in this. So it's really becoming more of an international scope type of a thing. More research, so the Arkin Lab published a sort of interesting paper with potential applications for metabolic engineering, this programming mRNA decay, which takes advantage of a native, this MASF endonuclease, which is targeted to specific regions in native mRNAs and chops them up. And what the Arkin Lab found was that if you delete you recode the target proteins that you want in enzymes that are participating in your metabolic pathway, you can reduce and shift some of those endonucleases away from degrading the proteins that you want and potentially improve titers. But what's cool is that it's actually a stable system if you do this. So they do a lot of the control theory behind the scenes to show that this has a feedback mechanism in place to get very stable regulation. The Baker Lab kind of just pulled another Baker Lab. So this time probably whoever went to SEED is going to talk a little bit about this. But probably the most interesting thing they have out here is this computational design of a trimeric binding protein to hemagglutinin, which is a cell surface receptor on cells that are infected by flu virus. So one challenge that people always run into when you're, for instance, making a flu vaccine is they make these like trivalent vaccines because we don't really know what the binding pockets are going to be every year. So what they did is they actually found that they can engineer these proteins de novo, as the Baker lab is wont to do, and they can get really, really good, if you look on the right there, the red line is when they use these proteins as a prophylactic, and it really helps the mice survive the flu better. An interesting paper published more in metabolic engineering. This was actually done in collaboration with ExxonMobil, engineering algae to be better lipid producers. So they found that if they just delete this one transcriptional regulator from the gene, they can turn the cells that look like this into cells that look like this, where 60% of the cell is actually making lipids. That's comparable to the sort of yields you see in urea, which is sort of the workhorse for making industrial lipids, but this is done in algae, so there's a lot of potential benefit that way. CRISPR news. For those of you that haven't seen this, this has been sort of a mess. It all sort of started out this last week in May. This lab in Iowa, I think it's actually more of a hospital, the Mahajan is the last author in this paper, publishes unexpected mutations after CRISPR-Cas9 editing in vivo. So they did a bunch of germline mutations to mice using CRISPR. Things seemed to work, and then they did whole genome sequencing and found that there were like dozens and dozens and dozens of off-target effects, indels, deleterious mutations all over the place, not where they were trying to mutate, and also not where computational design algorithms would predict the most likely place that they would get off-target effects. So this was quite bad, and once this paper hit, stock price of Editas dropped precipitously. All the CRISPR biotech companies started dropping precipitously. People didn't really quite know what to do with it. I guess it's recovered a little bit since then, possibly on the strength of the resulting paper that got published on bioRxiv just about 2 weeks later, where this is from Editas saying basically you guys screwed this up. This is a really fun paper to read, honestly. They basically said, you guys suck at what you do, we don't believe your results. There were definitely some suspicious things in the original study, so they only used 3 total mice. They had 2 treated mice and 1 control mouse, and the 2 treated mice were like relatives of each other. They were siblings, where the other one wasn't. They said the sequence depth wasn't good enough to get these to draw the conclusions that they had, and so on and so on. You can sort of see George Church just laughing at them. Again, if you Google George Church laughing, that was the best I came up with. They published a follow-up to the rebuttal now, and that's also on bioRxiv, where they're essentially saying, yeah, it wasn't necessarily the best experiment, but we stand by our data. We think this is real and something that people need to be aware of. I love the end of it. Overall, we are optimistic that some form of CRISPR therapy will be successfully engineered to treat blindness. So we're all on the same page there. And then this is George Church with black eye. It turns out if you Google George Church, this is like the 5th image that comes up. So, I saw that picture and I was like, this is going in the presentation somehow. All right, back on actual science. So the Silver Lab published a really cool paper talking about more of this gut microbiome stuff, engineering E. coli that can actually function as sensors inside of the mammalian gut. So what these bacteria are is they're able to sense a marker of inflammation called tetrathionate, which will switch on a genetic circuit, essentially a toggle. And what they find is that mapping the genome of these organisms after a month, they're able to maintain that toggle switch being active, which is really cool because you can potentially use this as a diagnostic tool or potentially even to deliver a therapeutic. A bunch of new cloning things happened. If you're interested in cloning, which I'm sure you all are, I thought the coolest one was actually this paper from NAR, which is essentially a new way of doing non-enzymatic DNA assembly, taking advantage of a lot of the techniques that people do when they do Gibson, but rather than sort of creating NEGs using exonucleases and then filling them in, what they do is they create like 2 sets of overhangs for every for every construct that you're trying to assemble, and then they denature them and then they reanneal them so that they can get the fragments to overlap like one after the other after the other. They finally get really great fidelity, comparable to Gibson, also really great efficiency for the assembly, and potentially could be a lot cheaper because you don't have to pay for all the enzymes that go into a Gibson assembly. And you had a bunch of papers out, so Colin in the Lux lab got this paper published in ECS Synbio on a split broccoli, which is a great way that people could potentially measure RNA-RNA interactions without the need of stupid proteins. Sorry, tough crowd. A lot of people in the Jewett lab here, Weston, other people I'm sure are similar out here. This paper on a cell-free platform for doing glycosylation just came out. I think it's a good preface to a lot of other work that's going on in that lab. Tolman Ursek lab published also in ECS Synbio about some new improvements that you can make microcompartments to improve the diffusion into the spaces, and potentially that would be really useful for pathway optimization, metabolic engineering type things. I caught this, it looks like a preface to a review that's got Devin from the Leonard lab on it on exosome delivery to breast cancer cells, so looking forward to seeing more in that space as well. I think that's all I have. Any questions? No one from online, please.
Not yet.
Please. Please don't do it. I can't handle it.
Oh yeah, that's easy to fix.
Patrick, do you want to go?
For your slides. Alright.
How bad did you tracking my slides?
Hi everybody, I'm Isaac, you probably know me. I'm going to talk for a little while about SB7.0, the 7th Great International Synthetic Biology Conference. I was not at SB7.0, I was in Ohio, it was in Singapore, literally the opposite side of the globe. But because it was the opposite side of the globe, it was 12 hours apart, and they live streamed the whole thing and took questions on Twitter. So I was able to just basically stay up all night, 3 nights in a row, and watch a lot of it. So it was really, really amazing. There were too many things to get into, a whole bunch of detail on, and a lot of the stuff that was presented there in terms of research will probably show up in like later newsreels. So I'll just talk mainly about one particular thing that has come up already, which is the creation of this biotechnology commons. And I want to give some context for this. Basically, one of the rate-limiting obstacles in synthetic biology right now is intellectual property. And that's because the intellectual property framework that works best for synthetic biology is one where you can take established parts cheaply and use them yourself, modify them, recombine them, remix them in a way that is sort of very easy and clean and doesn't involve tons of lawyers. Now the problem is that the IP framework for biology now is based on sort of the first wave of biotechnology, where it was very difficult to isolate a biomolecule that does something interesting, like a protein. It was very, very difficult once you figured out that interesting protein to find its gene sequence in the genome. And then it was even more difficult to take that gene sequence out of the native organism and put it into a useful model organism to sort of start making tons of it and selling it as your biotechnology product. And because all three of those things were really hard, it made sense to be able to patent individual biological parts, and even to patent organisms that contain a single useful biological part that's been inserted unnaturally into it. And this is still sort of the basis of the entire biopharmaceutical industry, the basis of Monsanto's agricultural products. So it works, the patent-based framework for biotech works well for that model, and it's makes a lot of money. But if you want to be able to make sort of more complex things out of a bunch of different parts that you remix, it becomes very, very expensive. And so Drew Indy and the BioBricks Foundation have been thinking about this for a long time. And they've been trying to make this sort of commons and drawing on inspiration from the open source software and the free software movements, where basically you publish your source code so anyone can use it, or you, in software, you actually use copyright to enforce that anyone who uses your code has to then freely share the code that they have created that uses your code. And so Drew Indy and the BioBricks Foundation and other people have been trying to do this for a while. The first iteration of that was actually the Antigen registry. They take all of the parts that the college kids make in the Antigen competition and they put them in the Antigen registry and they transfer it through this material transfer agreement that ensures that nobody will get sued for using those parts if they get them from the BioBrick registry. And so this is the evolution of that, but it's sort of a massive step forward because instead of sort of slowly, incrementally adding in whatever parts college students working on over a summer produce, this is creating a gigantic sort of 10,000 gene commons, and it's also using a new material transfer agreement that allows distribution not just to academic centers, but also to small community labs, to companies, and enables people to then commercialize products that they make from recombining these parts. So basically, it's really, really cool, and I am super excited for it. And I think one of the coolest parts of it is that we get to choose what goes in it. They're going to create a forum. It's not clear how the forum is going to be set up. It might be a subreddit, actually. They have one reserved already. It's called Bionet, where people will upvote suggestions for what should go in the registry. And the ones that get upvoted the most will get synthesized first. And BioBricks will pay for the whole synthesis and will basically distribute these genes to anyone who wants them free, plus like maybe the cost of shipping. So can I just get a quick head count? How many people will be interested in helping to brainstorm what should go in this registry? All right, decent number of people. Cool. Yeah, we're definitely going to be doing that. So we'll find you. Yeah, that was the coolest thing I saw at SB7.0.
So one of the sessions at SEED this year, which is one of the big synthetic biology conferences, was on T-cell engineering. So the current state of the art in T-cell engineering is something we call chimeric antigen receptor modified T-cells. So in this design, you use an antibody on the outside of the native T-cell receptor to target antigens that are specific, or mostly specific, to tumor cells. When that antibody binds to the antigen on the tumor cell, it engages the receptor, causing the modified T cell to kill this cancer cell. Or at least that's the theory. It works great for blood-borne cancers, and we're having some trouble translating it to solid tumors. It's got some new both medical and engineering challenges for the field. So the first big one is that one antigen might not provide enough specificity. And so you can imagine that anything a tumor cell is going to be expressing, something else in your body might be expressing. And so this leads to your T cells killing something maybe in your liver or your heart, and that's not great. So we call that on target because it's binding the right antigen, but off tissue because it's not in the tumor. And so everybody is kind of working on this problem, but the two big talks this year were from Wilson Wong's lab at BU, and he uses this, what he calls a Supercar system. This is pulled from his patent on it because it's not quite published yet, where the receptor itself is capable of Boolean logic. Instead of doing it at the transcriptional level, like most people do when they multiplex receptors, and is difficult, he's done it extracellularly through some cool protein engineering tricks with leucine zippers. So he's made a bunch of them, but in this one example, there's an AND gate. So you have your first antigen here and your second antigen here, and they have antibodies attached to leucine zippers. And the theory is that the leucine zipper interaction with the T cell receptor is so weak that you need both of them to engage the receptor and hold it clustered so that the T cell can activate. The other one is out of Yvonne Chin's lab at UCLA, and she uses this strategy called covert. So tumor cells also upregulate proteases and other genes. And so their theory was maybe we could use this property instead of looking for more antigens. And so the way this works is the enzyme that kills cells that is delivered by the T cells is something called granzyme B. And so they took a form of granzyme B that's inactive and separated the inactivating domain, the inhibitory domain from the rest of the enzyme. They put it together with a substrate for a protease that is overexpressed in tumor cells. So the theory is if this gets delivered, if a T cell tries to kill a not tumor cell, the granzyme B goes in there and it can't be activated. But if it tries to kill a tumor cell, it goes in there and is activated. And this works a little bit. It was published in ACS SynBio this past year, and they're currently actually working on replacing like the native granzyme B using Cas9 with their engineered one. So the second challenge is the immunosuppressive tumor microenvironment. It was really impeding T cell function. So you can imagine the tumor doesn't want to be killed by T cells, so it produces all sorts of molecules that are like, hey, don't kill me. And it works pretty well. So a lot of people have worked on this, and again, Yvonne's not published this work yet, so all I have is her, the description of her patent from the UCL website here. But basically, they've been able to design a CAR against one of these immunosuppressive molecules, so if the tumor tries to signal, hey, stop killing me, the T cell interprets that as, hey, kill harder. This works really well. It's cool. Cool stuff. And the last one is this new one called Antigen escape. So this is a normal biologic phenomenon. If you apply immune pressure against a specific antigen, cells that express less of that antigen will survive and become the dominant population. It's a natural selection theory for tumors. And one place we really see this is in leukemias and lymphomas, where there's two antigens that these B cells express. They express CD19 or CD20. And the functions of those two molecules are slightly redundant. So if you design a T cell against CD19, which is the state of the art for B cell leukemias, you wipe out your CD19 positive B cell population. The problem is, then these tumors recur, but this time they're CD20 positive. So imagine if you had a T cell that was capable of killing both CD19 and CD20 positive B cells, that this would prevent the immune escape phenomenon. So this here is a normal CAR. It's got one antibody attached to the outside, so it can recognize one antigen. This is a bispecific CAR, so it's got two places where it can bind antigen. So it can bind CD19 here or CD20 here. And in a mouse model of leukemia, where they study this population relapse and immune escape phenomenon, it was able to extend mean survival by about a week using the bispecific CARs as opposed to the monospecific one. And so this is actually probably enough data that she's going to take this into preclinical trials, which is pretty cool. So that's it for me. Thanks. Yes, Isaac.
Are there other tissues in the body that produce like these immune suppressive signals that the chimeric antigen cells might?
Yes, and she has them multiplexed in a way that you need both the initial antigen and the second immunosuppressive signal to get full activation.
Cool. So another great talk at SEED was a keynote from David Baker. Of course, any talk about David Baker is going to be, every slide is something that either is about to be published in Science. So it's a little humiliating in that way. But essentially the takeaway is that really what synthetic biologists have been doing, which is taking proteins from nature and trying to repurpose them for what we want to do, it's very primitive and in his words sort of Neanderthal-esque. What we really should be doing is what they're doing, which is designing proteins specifically for our purpose. So a few highlights from what they're working on. They have, as Adam already stole my thunder a little bit, these trimerized HA and botulinum toxin binders. So you have these trimers that bind to 3 different antigens of the toxin. You can get really good neutralization of flu and other things. And then another really interesting thing they're talking about is this proteins that are held together by central arrays of hydrogen bonding networks. And so this is sort of similar to, or analogous to what you see with DNA, where you have Swanson base pairing. But with this, you can now add multiple strands. You can have 1, 3, 4 strands, or as many as you want. And they're doing some cool things with this, like adding hydrophobic patches so that these things incorporate into the membrane. You can basically start designing like transporters and stuff like that with this type of strategy. And they also have a lot of cool things like allosteric protein switches that cause these trimerized binders to switch to a binding state just by adding a peptide key. And they're doing some interesting work designing these nucleic acid proteins and trying to evolve there. Another great talk was about synthetic epigenetics. This was from Ahmad Khalil at BU. The essential idea here is to create memory by using chromatin regulators to basically write and erase DNA M6A methylations. And these things can be engineered with zinc finger binding domains that recruit your modification enzymes to specific DNA sequences. You can add in M6A modifications or remove them, and then engineer readers that essentially translate those modifications into a M6A dependent gene expression activation, stuff like that. So that's just an example there where they have background where you see low gene expression, and then when you add your chromatin modification, you get increased expression from that EP16. That's about it. Include anything on there?
Hi guys, I'm Weston. I'll be the last SEED speaker, so I want to go over a few things that I saw that were particularly exciting. One was a presentation by Oxitec, so a lot of you are probably familiar with them, but they are basically interested in the genetic control of insect pests, in particular mosquitoes, and they're actually the furthest along in this. There's several companies who are attempting to do this with things like gene drives, but Oxitec is actually getting quite far in that they're actually doing trials at this point. So it was a good update to hear from industry. The stuff may be available, but I haven't been able to find that much on the news. But what really struck me, and so they're out of Oxford, and so they're targeting out of the UK. But what struck me is that it's not just mosquitoes; they're actually targeting like a whole slew of various agricultural and public health pests. And so these are like medfly, olifly, like all kinds of moths, all different kinds of mosquitoes, and like sable flies. So it's actually they're trying to build this entire company around basically suppressing these populations of pests. And what's interesting about their technology is that it's inherently self-limiting, and they made a big to-do about that, so which is accurate. So I think that this might be like the synthetic biology solution we need right now. And so the idea is that they engineer male mosquitoes and they grow them actually in the presence of, I think it's actually TED, right? It's actually like a TED repressor, and they grow them in the presence of that. And so they're fine when they grow, but then they release them into the wild, and then they can mate with females, and then their offspring don't have that TED environment, and then they basically die during development. So and females only mate once, so this is a way to suppress the population. But if you stop releasing mosquitoes, then your population will bounce back, hypothetically. I mean, population crashes are a thing, so I would argue that it's not entirely reversible, but but certainly not anything like gene drive. So it's important to know that like these kind of technologies are out there, and they threw out some pretty impressive statistics. Like they did a Cayman Islands trial in 2009, which I didn't even know things were happening that early. 96% suppression of the target mosquito population. A lot of this is with the yellow fever mosquito. They did a trial in Panama. They've done a number of trials in Brazil, over 90% in all cases. And they have production facilities where they make like 60 million of these male mosquitoes a week, which is like just a pretty amazing technological marvel. It sounds like counterproductive, right? But and then I guess really critical for the male mosquitoes thing, or for the mosquitoes thing, is that you can sort males and females by size. But but that's not true of all insects. So they actually had to come up with this like workaround where instead they target the females, and then the female offspring die also later. That one, so they had an interesting genetic workaround for that. But then they also have like a bunch of public education efforts. There's like Friendly 80s or something like that. I don't actually know how you pronounce that name, but they have like they showed like pictures like in the middle of Brazil, like these booths with like this like mascot who runs around and like tries to be a friendly mosquito. It seems like mixed signals to me. But yeah, so they are definitely working on that. Monroe County, which I think is in Florida, I think it's actually the Florida Keys, has approved basically 31 out of 33 precincts or whatever have approved the use of Oxitec technology to fight Zika virus. So this is coming to the U.S. Soon, so that's cool. So we talked a little bit about Twist bioscience, data storage a little bit, but what's fun about conferences is you can actually like corner people and like ask them questions. So Emily LaProuse was there, and I actually walked up to her and was like, hey, we're from GeneMods, and I mentioned how a few members are really interested in using their BioNet system, and then what I really wanted to ask her was, why do DNA data storage? It still hasn't really made sense to me. Like, I'm as much a synthetic biologist as the next guy, but seriously, I could understand if you want to shoot the Library of Congress to the moon, might be helpful, but that seems like a niche application. So, she actually threw out some really impressive ideas, and so this is really helpful for me when I think about this. So apparently oil fields, when they do seismic surveys of them, those surveys are worth like hundreds of millions of dollars, okay? And then they keep those oil fields for like hundreds of years. So they need that technology, or that information as a backup to last like hundreds of years. And so if they would lose that when hard drives get corrupted, if that gets corrupted, then like you have to resurvey your entire oil field. So they keep mining the same field. So apparently that's like itself a multi-million-dollar industry. Medical record backups are actually legally required, and if you like would lose those, you're actually breaking the law, so there's good reason to have several backups for those. Apparently, she talked to James Cameron, who was making Avatar 2, which is coming out, and he wanted the audio files, like the really rich audio files that it takes to make movies, right? Actually, gigs and gigs, right? And he's like, okay, team, get out the Avatar 1 audio files, and they're like, we don't have them, because their hard drive crashed. So he was really pissed off about that, and now wants to use DNA data storage, I guess. It's kind of fun. And then there's a big market in defense-related data storage. So DNA, because it's physical and like not connected to the internet in any way, can't be hacked. So that's kind of cool. And then it isn't destroyed by like electromagnetic pulses. So you can imagine, like, God forbid, there's like a hydrogen bomb, like half the United States has all the hard drives are gone, right? So your DNA would not be affected by this. It's like in a lead case. And so you still have to get it to a sequencer, but assuming you can get it there, get this cataclysm. But anyway, it's an interesting idea. They'll probably sell some stuff that way. Microsoft is certainly investing like millions and millions of dollars. So I hope Microsoft isn't stupid, but it still remains to be seen. But anyway, just like to put some meat behind that, that was a lot of fun talking about that. And they have these cool acronyms. So apparently there's like a whole class of data called worse and warn that like basically never gets read, but you still have to have it because reasons. The other thing I wanted to highlight was a few talks I thought were just like really good. They're like not necessarily what you would call like the keynotes, but I thought they were really great. One is on, so there's a whole section on engineering the microbiome, which I think we're getting further on than I thought we were. So there's a group from University of Minnesota who's working on microbial delivery of antimicrobial peptides. So you engineer a bug, then you to produce antimicrobial peptides, then you ingest that, and then it like shoots them around to the drug-resistant bacteria in your gut and can therefore treat. And that's cool because antimicrobial peptides can be much more surgical than standard antibiotics. So, and also a lot of them don't have resistances yet, and there's a lot of them, so you could use that. There's also a really big market in the poultry industry because you could be antibiotic-free but use antimicrobial peptides. It's a cool trick. And you're not modifying the chicken, but you're putting in a modified bug. Fun. So that'll probably hit Whole Foods at some point. Or Amazon. So SynLogic also had a cool talk. They have a whole variety of metabolic disorders that they're trying to engineer bugs to treat. Usually you ingest these bugs and then they inhabit your gut somewhat. The issue is colonization, and that has been the issue for a long time, but they're working on that. And it can basically do what your body can. So whether that's producing butyrate or like digesting arginine or like a variety of other things, it's kind of a cool idea. And then they have this whole thing, it reminds me, does anyone remember like this paper where they were like, we can treat cancer by injecting like E. coli that go kill the cancer, right? And you read it and you're like, that's called sepsis. But they're trying it, so apparently they're really excited about it. And I guess they say that their E. coli like only live where the tumors are, so they're friendly and all this stuff. It's still gonna be a while before other people like shoot live bacteria into my bloodstream. But anyway, definitely they have a large arm in that area. And then what I actually thought was the best talk, and unfortunately it's still unpublished, but just an incredible talk by Michelle Chang at UCal, Berkeley. And it's basically this idea with metabolic engineering to bioorthogonal chemistry. So there actually are some bugs that produce alkyne amino acids. So if you're familiar with like click chemistry, so these are alkynes and azides, you can do click chemistry with those. And what would be really fun is if you could make your bugs produce your bioorthogonal chemistry. So have like naturally occurring non-standard amino acids, right? So in this way, you can produce actually your non-standard amino acid, which could be useful for bioorthogonal chemistry, but have it be made in the bug rather than like adding it in the media. So anyway, she did an incredible, basically like really chemistry heavy, really mass spec heavy exploration of this pathway, and I do think that they'll be able to put it into E. coli soon. So, so it was a lot of fun. That's all I have. Any more questions for me or the other speakers? Yeah.
Were the first two talks just coli-based, or are they looking at other microbes?
Yeah, so the workhorse here is Nissl. So E. coli, Nissl, 1917, isolated World War I-ish time period. So yeah, that's what they're using. And it seems like they're kind of settling on that chassis at this point. But they're using it to treat, salmonella is one of the things that they like treat and various other bugs. But, yeah. Cool. All right. Oh yeah.
So for Oxytex, if people who haven't seen it, on their website they have these amazing like promo videos. It's like technology their food, so
Cool. Yeah, it's really fun.
way animations, mosquitoes.
Really? Awesome. Yeah, I just want one of those like costumes, right? Like their mascot. It'd be a great Halloween costume. Oxytex mosquito. All right. Well, thanks guys. Stay tuned for more updates. All right. Stay tuned for more updates from GeneMods. Definitely listen to the podcast. It's incredible. And then we'll have a meeting next month. So thanks. No, I was really worried. I feel like that also like if you're doing that.
Backfire.